Welcome to the 1-2 podcast for biology. And this section is on how scientists work, and we're going to talk about how scientists do their work and at the same time look at a rather famous set of experiments and use them as an example to understand. So, start off. How do the scientists test an hypothesis? The main point here is that they use an experiment, and the experiment should only have one variable changed, and everything should be kept unchanged or controlled. So let's test the hypothesis and look at how they do it. And there are steps that scientists use. First, they ask a question. Then they form a hypothesis, and remember, a hypothesis once again is a prediction that is to be tested. They set up a controlled experiment. Remember, they only want to try to change one variable at a time. They record the data or their observations, and then they analyze these observations. And then from there, they draw a conclusion from the observations. So here's the question that we're going to use to look at as an example, so we can look at these steps, these 5 steps in designing an experiment. And the question is many years ago, people wanted to know how living things came into existence. So they asked, how do organisms come into being? That's the question they asked. So on to the next step, forming hypothesis. One of the first ones was the hypothesis of spontaneous generation, which basically means that life can just sort of happen. It doesn't have to come from some other living organism. It can come from non-living matter. Some people believe that maggots just magically appeared on meat. So, a scientist named Reddy proposed a hypothesis that maggots came from eggs that flies laid on meat, and he went about testing this hypothesis. Let's go on to the next step, setting up a controlled experiment. So, first thing we need to do is define the word variable, and it is something that can be changed or controlled. The variable that we deliberately change is called the manipulated variable, and then the variable that that responds to this, that you observe that changes, is your responding variable. So let's look at Reddy's experiment. What he did was he took 2 jars and he covered some of the jars and left other jars uncovered. And his observation is that the flies land on the meat that is left uncovered, and then there are maggots. And his hypothesis then is that the flies produce the maggots. The controlled variables of the jars, the type of meat, where they're located, the temperature, time. You put these things right next to each other. But your manipulated variable is the covering on the jar, the gauze. And the responding variable is whether you see maggots or not. So maggots did form on the meat that was uncovered. So what his conclusion was is that maggots form only when flies come in contact with meat. Therefore, spontaneous generation is a myth. It doesn't occur. So let's go to the 4th part of the method, recording and analyzing results. Scientists keep written records of their observations or data, and sometimes they use drawings to record certain kinds of data. You've seen all these before. Scientists will use computers, and online storage makes it easier for the researchers to review the data. It makes it easier for people to keep massive amounts of data, and also to share the data with other people. But let's get back to the procedure, drawing a conclusion. Then what the scientists do is they use the data from the experiment to check the hypothesis. They go back and say, is the hypothesis correct? And they draw the conclusion from it. And Reddy, his observation showed that the maggots were produced where there wasn't gauze, and where he saw flies land. So his results supported his hypothesis that it was produced by the flies and not by spontaneous generation. Now, repeating investigations. Scientists repeat them just to make sure they're accurate. And there was a guy named Needham who didn't believe Reddy's results, and he said, well, there's got to be a way that spontaneous generation occurs. It's if you have the right conditions. So what Needham did was he sealed a bottle of gravy, and then he heated it, and after several days the gravy was forming with microorganisms. Okay, that seems strange enough. So he concluded that the organisms came from the gravy by spontaneous generation. His argument is that he heated the gravy enough that it should be all the microorganisms are killed. The problem was he didn't boil it. So a guy named Spallanzani boiled the gravy. If you notice, he used 2 flasks, and he left one flask open, and then he sealed the second flask and compared those results. And now you can see that the one that was open was teeming with microorganisms. The one that was boiled and sealed was free of microorganisms. So he refuted Needham's findings. Now, Louis Pasteur was a very famous scientist, and he basically is he went a little further than these 2 guys, and what he showed that all living things come from other living things, just not a very specific hypothesis. He once again broiled broth, which is essentially something that organism has nutrients in it. It's a liquid that nutrients are in, so that a microorganism could live in it. And he found that he used a curved neck on his container, and what he found was broth is free of microorganisms for a year. And the problem was that the microorganisms could not get through the neck of the bottle. Then he removed the curved neck, and once he did that, then the microorganisms could get in and breed inside the broth. And this Pasteur's work helped the wine industry, the silk industry. It was and from there they figured out how infectious diseases occur. So his contributions were very important. In fact, one of the processes they use to get rid of microorganisms is a process that it's called pasteurization. You probably heard that your milk is pasteurized. Now, these are just the little sides. When experiments are not possible, sometimes you can't actually do a lab, do a controlled experiment. What you'll do is you'll just observe, for instance, wild animals in the wild, and you can learn a lot just from that without actually having a controlled experiment. And also the ethical considerations prevent some experiments. We don't test harmful chemicals on human beings. In fact, now our ethics are changing so that we're not testing harmful chemicals on laboratory animals in a lot of cases. So, and the point is that you can still add to scientific understanding without using the classical controlled experiment. If there's other tricky ways of doing it where you still can get valid results using alternative methods. So how does a scientific theory develop? Let me go back to that. Is the evidence of the investigation builds up, a hypothesis may become so well supported that scientists consider it a theory. Now you might be saying, well, a theory is not a guess. A hypothesis in many ways is a guess. A theory is an explanation, and it relies on a lot of different experiments. It's broad, meaning it covers a lot of different examples, a lot of different aspects to explain some concept. And one other thing to remember is no theory is considered absolute truth. As time has gone on, we've refined many theories. There's a theory of how the universe started. The theory of evolution has been refined. There's many theories that are changed, because as you find new evidence, you can revise it or even replace it and I think a lot of them are refined, meaning making them more accurate. Now, once again, we use the word theory, and we're talking about the theory of evolution. And once again, I have to stress that the word theory in science is not the same as the word theory in our normal layman's terms. A theory in science is an explanation that has been backed up by evidence, whereas a theory in layman terms is more of a guess. Theory, almost why the Mets are so pathetic every year. I have my theories. I don't have any evidence to back it up, but therefore it's not a scientific theory. It's just a guess. I'm sorry, and I got to point out this website, www.notjustatheory.com. It puts the idea of a scientific theory in great and really easy to understand terms. On to the quiz, and I'll just run through the quiz questions and pause at them. You can look at them and try to guess at them and then see the answer. And that's the end of the section.